first would like to thank Sven and Didier to be able to bring together people in such a charming way. I think this is one of the most charming senescence or aging meetings I know. And also very informal and very refreshing exchanges. So today I will try as quick as possible to convince you that you can try to address the inflammatory facet of SASP. So senescence, was it something you were thinking about? What is it? We all know the classical definitions, and we also know that in fact the only thing that really changes is the shape of the cell. So at some point when I presented for the first time in a Gordon conference some 28 years ago my first results on senescence to Heiflich, he was saying, "Oh, is it reversible?" And I said, "Is it reversible? I don't know." So I started to look for another definition of senescence, a reversible one, although I didn't exactly understand what he meant. So I was working on endothelial cells and was noticing that upon a TNF stimulation, a pro-inflammatory stimulation, cytoskeleton, I like the Spanish guy talking about rhodamine, there was a heavy actin proliferation that gave rise to stress fibers. And these stress fibers were co-distributed with a high expression of adhesive molecules. Now, this is a perfect reversible process in the young cell, because from an inflammatory phenotype you go back to a not inflamed phenotype. Yet, in the senescent cell you have previously characterized as such, this process is not reversible anymore. So, I was progressing with some hypothesis and saying a senescent cell is something in which there are stress fibers and a hyper expression of pro-inflammatory cell adhesion molecules. Why are they pro-inflammatory? Because in fact they sustain further inflammation. And as we know since a while, they also are interesting not only the vascular department, but also other departments in other organs, and especially the stem cell department. Now, if inflammation favors adhesion, adhesion is favoring inflammation. And in fact, these cell adhesion molecules are fitting into the integrin system that is expressed by the activated leukocytes and is draining them into the site of injury, but eventually also some cancer cells. So this system is a punctual system that has to act quickly, efficiently, and be reversed. At some point, it is not reversed anymore. It is entertained by pro-inflammatory cytokines or other pro-inflammatory signaling. In particular, as Sven talked about extracellular matrix, I would like to acknowledge this presentation saying that indeed, not only extracellular matrix is extremely important, but also following pro-inflammatory stimulation, the internal tension and forces in cells are modified and are creating the possibility to change partnerships in cells, and thus arise the arm, the intracellular arm of cell adhesion molecules in order to fit them to circulating leukocytes. So the first checklist at this point to identify the molecule that would be able in a non-toxic way to perform an anti-inflammatory and anti-senescence activity would have been to control immune regulation at vascular level, and we were able to inhibit endothelial adhesion molecules to a very high extent, 80%. And also to modify cytoskeleton, which is at the base of the change in morphology in the senescent cell. And this was done through this molecule I will characterize further, inducing a heavy actin depolymerization upon TNF stimulation. Then I met Aubrey, and in 2007 he invited me to give a lecture in Sense 3 in Cambridge. And there I started with what I had, these first results, to characterize that in fact, eventually, it would have been possible to reverse senescence if we were able systemically to inhibit TNF-alpha and interleukin-6 that were promoting the senescent phenotype in organs. And indeed, as you all know, the SASP hypothesis of Judy were indeed at the time in the way of publication. So what is this molecule? In fact, it's a molecule that I identified, and I even got an award from the Ministry of Research in France for this, and which means a lot of money to do research, in the rainforest at the boundary between Vietnam and China. And these molecules were collected, these plants, each year by ethnobotanists of the University of Hanoi. So when I identified the molecule out of 100 plants, which means 200,000 molecules, I only looked at 2,000. This was what immediately came upon was that they were potent inhibitors of farnesylation of Rho. Now, Rho is interesting because of its integrin engagement upstream, and then downstream because it's actin cytoskeleton remodeling. So we were exactly in our scheme. We didn't know, we just observed. So the second thing we said, there is a relevant pathway. Let's look if it's inhibiting NF-kappa B. And yes, in a fibroblast model, it was potently inhibiting NF-kappa B. And we did this work with the IGBE group in Naples. And finally, it was inhibiting neoangiogenesis. And this for us was a very important point because of the link between senescence and cancer. But the story is just beginning. Does it work in vivo? So we started, first we had seen some with our group in the Charité. Professor Schulzke had a fantastic model on barrier function. He's a gastroenterologist and swears on barrier function. So we looked at, in fact, we were efficiently restoring barrier function in an enterocyte model. So collecting all these evidences, we started a rat colitis model in which we showed histologically and systemically, this is circulating TNF-alpha here, whoops, there, that we were able to equal the ibuprofen effect, simply it was not toxic. So in preclinical texts, we continued to show and demonstrate that they were able to vehiculate wound healing without angiogenesis, which is a quite interesting story. And always we documented that here we had a total inhibition of the major pro-inflammatory cytokines in the serum. Now, tissue repair models are sometimes conceived to mimic pathologies. So in this case, it was psoriasis and atopic dermatitis. And we were able to characterize that indeed it was able to repair. And in this case, we also could see that the metabolite of D-limonene was in fact very interestingly active, which is not the case in all the competences of D-limonene. Of course, again, in the tissue, we found adhesion molecule inhibition, in this case, P-selectin. So having dealt a lot with animals, which I was not used to because I had lived my life with my cells, we were able to observe curiosity and ludic behavior, and, as it was addressed this morning by several authors, exploratory behavior. So we thought, what's going on? What has this behavior change to do? And indeed, we met a fantastic ethologist who had a functional observation battery. And here we were able indeed to identify the incredible difference between a frozen rat and a rat in a total different behavior. We have also lots of films of these rats which were suddenly playing with a wheel, checking for the neighbor, and shuffling. Instead, the non-treated rats were quite irritable and frozen in the coin of the cage. So we published this again in Rejuvenation Research. Thank you, Aubrey. He's not here, but he was very kind to give us so much space. We continued for human studies. And in human studies, we saw effectively in a psoriasis model the capacity to engage the famous stem cell compartment and wound healing and anti-inflammation. But what we are interested really in is this life quality index. And this was greatly ameliorated. So when it came to the European project Ristomed, for the first time we were challenged to use something else than these Indo-Chinese molecules from these plants, because this was much too exotic. So the commissioner said, it's healthy aging by nutrition, so find your molecule in something you eat. So my brilliant PhD student, Anthony Marconi, spent 36 hours on the web and found that in the skin of the orange, this molecule was represented 99%. So we were quite happy, and we tried to press cold the skin of the orange to get a high concentration of this molecule. And this was administered to 240 patients in a study, which was a multi-facet study, Berlin, Rome, Bordeaux, and Bologna. And we obviously could demonstrate that interleukin-6 was efficiently inhibited. These people were 65, 85 years old, and they were not sick, but still they were depressed and inflamed. So we also found out that some interesting cardiometabolic markers, here fibrinogen and insulin and HOMA-IR, were efficiently inhibited. And again, for us, very importantly, that the depression score, specifically in low inflammation, was quite ameliorated with a slight gender issue, specifically in women. We made a paper on women depression different from male depression. So, has AISA and mood something to do? AISA is an acronym for anti-inflammatory senescence actives. Is it a dopamine story? Why do I raise this stuff? Because when I presented my first results in Cambridge, there was a guy from California saying, oh, your story is just rohypnol, nothing new. And I said, yes, it's new because rohypnol has a narcotic effect, whereas we have a slightly euphoric effect, which is the whole difference. So as you know, dopamine is declining with age. So motivation and just fun is decreasing. And just 2 months before my patent, a Japanese professor happened to file a patent on in a trial cared of by Nestlé, showing that D-limonene was able to raise, importantly, dopamine levels in the brain in a mouse model. So now we knew why the behavior of the rats were so changed. So So the intermediate checklist was transcription factors inhibition involved, yes, NF-kappa B inhibition, and we all know it's one of the pathways driving the SASP. Secondly, in preclinical animal models and clinical models, we could assert that inflammatory cytokines were taken care of. Finally, that we had important behavior changes and mood changes under non-pathological stress, I insist, non-pathological stress. So fighting inflammation to reverse senescence could in effect be something at some point that one could think to be able to reverse into a rejuvenated phenotype. Why not? And some evidence of this is for just a summary of what we did, only introducing the next chapter. But if you look at it, there is some coherent effect that is talking about something that is regulated by something, and this something you will understand in a while what it is, and it's part of our deliverables of the next 2 years. We will look at epigenetics. We will look at stem cell niches, because a brilliant group of Stanford just showed that dilimonin is able to protect stem cell niches when submitted to toxic aldehydes, and microbiota. That's the cue. So microbiota is sensitive to diet and stress. I have 2 minutes, I'm done. We already shown some capacity to inverse the Clostridium bifido ratio, always in the ristomet. But microbiota is talking to epigenetics and immunity in a constant dialogue. And so here is the cartoon that takes it all together. Here you have the neuroinflammation transducing into cortisolemia that is destructuring. Remember the cartoon where I showed the barrier function restoration. Here you have the leaky gut that is reinforcing the pro-inflammatory tendency. And here you have the most important thing, which is the epigenetic contribution of food or food components, because prebiotics and probiotics can create what are called the psychobiotics that are indeed generating the whole anti-inflammatory scale-up in order to contrast this other cascade. So ultimate goals of anti-inflammation, anti-senescence strategies. Aging can be delayed. This is something we know. But we think it's reversible. And in fact, these people apparently think the same. Anyway, they stay with the hypothesis. And next year, I will get a book out that I will be able to translate in English, I suppose. And the L of inflammation is red. I don't know why. And this will be for the large public, a book who is explaining all these quite short-cut passages I have fed you with this afternoon. Thank you for your attention.